Welcome to the video for chapter 3.3. 3.3 is all about bright field or light microscopy. Okay, as I said, chapter 3.3 is all about bright field or light microscopy. So our section objectives are you're going to be able to explain how to use a bright field microscope. You're going to be able to describe the different parts of a microscope, and you're going to be able to describe how we observe an object in focus by using these compound microscopes. Now, there is a disclaimer here on the slide, very big and in yellow, so we don't miss it. Okay, so if you read this section in the book, it goes into a lot of detail that we just don't need for this course. It goes into what parafocus or parafocal is. We aren't going to use that. So we're going to focus on the basics. So the stuff that I talk about in this lecture and the stuff that we talk about in the lab. So even though the book has a lot more detail, we don't need to focus on that detail. Okay, so let's talk about light or bright field microscopy. So we've talked about the two parameters that all microscopes function by, and that is magnification and resolution. Now, when it comes to magnification, this is the heavy lifter of the lenses. Okay, so lenses are going to be curved, and that curvature is going to determine how much light we can capture with that lens and how far we can magnify. Okay, so the greater the curve or the more aggressive the curve, the more light that we can catch and the more magnification we can get. Okay, so we have a lens called the objective lens. This is always the lens that is closest to your slide. So it's the one that's directly above the specimen. Okay, so we have four types of objective lenses on our microscopes that we're going to use in the lab. We have a 4x, this is called the scanning objective. We have the 10x, which is called the low power magnification. We have the 40x, and then we have the 100x, which is the high power magnification. So the difference between all of these objective lenses is the curvature of the lens. So if we look here at this lens, it has a pretty gentle curve. So it's not going to catch as much light. Okay, and because it doesn't catch as much light, it's not going to magnify the image as much. Okay, and it's also going to have some pretty poor resolution. So if we have two objects here that are close together like this, okay, we don't have enough space between them and we don't have enough magnification or light intensity to resolve the two and tell them apart. So we don't get really focused. We don't get a very crisp, detailed picture. Now, when we increase objective and increase magnification, our curvature increases. So if you look here at this lens, it is more aggressively curved than this lens up here. And because of that, it can gather more light, which means we can get higher resolutions. So we can look at the same two objects from underneath the 10x, and we can actually distinguish them as two separate things. Okay, there are some challenges that come with the 100x magnification. One is that the light is so curved that sometimes our lens can't catch it, even though it's capable of catching lots of light. It's also very, very close to our sample. It gets a little scary that way, actually, especially for your first time if you use the 100x. So those of you who took biology 201 or just regular high school biology, when we played with microscopes, the highest objective lens we let you use was the 400x. And that's because of how close the 100x actually gets to our sample. I have had students in the past break slides with the 100x objective lens, but we'll kind of slowly transition you into using the 100x, and we'll teach you how to use it correctly. Okay, so one of the other things that help is the fact that these lenses are made out of glass. That means they have a high refractive index. They have a greater refractive index than the air outside of the objective lens. Okay, so this refractive index is also going to help us catch more light and increase our magnification. But once we get to 100x, we can't catch all the light. And sometimes with the 100x objective lens, we really can't see anything because we're not catching enough light. So what we have to do is we have to use a special trick that we'll talk about here in a second. Okay, so we've kind of talked about resolution. Resolution also has to comes with the curvature of the lens. So resolution is how we're going to distinguish between two objects that are close together. So we can have high resolution where we can see more details and everything's crisp and clear, like this sheep here on the left. Or we can have low resolution where we can see the two objects, but we really can't tell the difference between them clearly, which is when we get the blurry image. Okay, it all has to do with the lenses here. Okay, so that special trick I was talking about is instead of using one lens, like we were doing on the previous slides, we are actually going to use multiple lenses. We're going to actually stack two lenses on each other. Okay, so our microscopes that we are using in the lab are what we call a compound microscope. Okay, so this is a microscope that uses multiple lenses in order to correct or compensate for aberration, meaning we use multiple lenses to magnify an object larger and with better resolution. Okay, we actually have more than two. We're going to talk about, in most times with microscopes, just two of the lenses, but we actually have three lenses in our microscopes. So if you look at this schematic, we have our objective lenses. Okay, so these are on a revolving nose piece, so we can flip between them so we can increase the magnification on our slides or specimen. Okay, but the other main lens that we talk about when we talk about compound microscopes is the ocular lens. Okay, so ocular means eye. So this is the eyepiece lens. So this is the lens that we look directly into right here. Okay, so this is the second lens that is going to increase our resolution and our magnification power of our objective lens. Now, the third lens that these have is what is called the condenser lens. So our compound microscopes in the lab are light microscopes. So they're always going to have this light source. This is going to be a light bulb. We have the diaphragm. Okay, so this is kind of an opening below the microscope that we can open and close to let differing amounts of light in. We have the condenser, which is going to start to focus. So this is the condenser lens. So this is going to take the light from the light source and it's going to condense it so it hits a small area of the slide. So usually we put the specimen right above this condenser lens. So we are shooting this light straight up and onto our sample. Okay, and that light can then pass through the sample, go through the objective lens, and then and then it will go through the eyepiece lens and it's going to increase the magnification. Now we have other parts of a microscope that we need to be worried about. We'll talk more about these in lab. We have the focusing knob. So in the schematic, it's up here. Ours are actually down here on the ones that we have in class. This is the stage or specimen stage. We have the specimen or slide clamps that will hold the slide down. Now, if you took Biology 201, we used mostly plastic slides. Now, our plastic slides didn't have a great refractive index, so they weren't as easy to get a clear image on. And also the slide clamp would go over them and not hold them in place. What's really great about microbiology is we've stepped up in the world. We're actually going to use glass slides. So the slide clamp will actually hold the slide in place. Okay, so these are all the different parts of the microscope that you need to know. This is the arm, this is the base, this is the eyepiece. Okay, we will be tested on this in both the class and the lab, so I would know these different pieces and what happens at each. Okay, so when we put two lenses working in conjunction with each other, something magical happens. Okay, so our light source always comes from the bottom. It's going to refract into the lens, it's going to catch the light, and it's going to focus it on our specimen. Okay, so this is the objective lens. We have our four objective lenses. Okay, and then we're going to use this ocular lens or eyepiece lens, and it's going to magnify the image even further when it catches the light coming through this lens and off of our sample. Okay, so this fact that we can use two lenses is going to increase our total magnification. And the way that we figure out what our total magnification is with a very simple formula. Okay, so total magnification is going to be equal to the eyepiece lens or the ocular lens times the magnification of the objective lens. So say I am using the 10x objective lens, Our eyepieces are always 10x. Our microscopes do have the option to switch it out for a 25x, but in this class, we are always going to use the 10x eyepiece. Okay, so we're going to take the magnification of the ocular lens, so the 10x, times it by our objective lens. So if we choose the 10x, it's going to be 10 times 10. So our total magnification of our microscope will be 100x. We need to know this formula. It will show up both on the in-class exams and in the lab. Now, we do need to talk about the 100x objective for a second, because in order to hit the highest magnification possible, we have to do a special trick with the 100x. So we talked about how the 100x doesn't always collect all the light it should. So sometimes we don't see a sample at 100x. We don't really see anything. So one of the reasons this happens is light's going to come through from the light source in our microscope. It's going to hit the slide. It's going to refract. And so instead of refracting so the lens catches it, it's actually going to refract and go outside of the lens. Okay, so this is why we can't see anything with the 100x lens. In fact, we don't ever use the 100x objective lens without a special substance. We actually use what is called immersion oil in order to, and this helps us use the 100x more efficiently. So right here in this previous example, when our lens couldn't catch this photon, this light, is because the refractive index of the slide is much greater than the refractive index of the air. So we're having a refraction here, so we're bending the light here, but not enough that we're going to hit the actual lens. So we're going to miss the lens completely and not get any image. Now, immersion oil has a higher refractive index than air. So what's going to happen is we have our light coming through the bottom of our slide. It's going to refract and bend. But when it hits the immersion oil is not that different of a refractive index. So what's going to happen is the refraction or the bending of this light is not going to be great enough that it's going to miss the lens, and the lens is actually going to be able to gather it, creating an image so we can actually see our specimen at the 100x objective lens magnification. Okay, what we do is we just put the drop directly on the slide, and then we put the 100x objective lens straight down, and it kind of creates a seal between the oil. The oil creates a seal between the objective lens and the slide, allowing for less refraction, so that's actually refracting into the lens and not missing it. Okay, this is something you will be tested on, so you need to know that our 100x objective lens is the special one that needs to use immersion oil, and you need to know why we use immersion oil. You need to be able to explain how immersion oil corrects the refraction so that the lens can actually catch light so we can see an image. Okay, so one of the last things we're going to talk about is how to focus. Okay, this is where the focal point comes in. If we want to have a crisp, clear image of our specimen, our focal point needs to be directly on top of our specimen on the slide. Now, if you remember back from Biology 201 and for regular high school biology, we do a lab at first where we create a wet mount. So we bring in a water sample and we take a drop of that water sample and we put it directly onto the slide, and then we put a cover slip over it and we put it into the microscope, and we start to observe any organisms that are in that drop of water. Okay, so by putting the drop of water on a slide, this is what we call a wet mount. We can do this with bacteria as well. We can get a drop of a broth sample where we've been growing microbes, and we can do this as well. So when we use a wet mount, we don't use stain, so it has very low contrast. Okay, so where we put the focal point, so how we focus the microscope, is very important about what we can see. Now we're going to go over to this image right here, and this is going to show us the differences between where the focal point is. So here in A, our focal point is below the sample. See here? Okay, we have our sample on our slide, the focal point's below it. What's going to happen is our sample is too close to the lens, And so it's going to be very blurry. So it's going to be kind of like this. So this is what we see here with our sample. It's just blurry. We can kind of see the microbe, but we really can't tell anything about it. Now, if we have the focal point directly on the specimen, what we're going to get is a nice in focus, crisp image. So we can see a lot more detail. And that's what we're seeing here in this micrograph, is we can see all 4 segments of this microbe very clearly. Now, if we have a focal point that is above the specimen, okay, that means that our our slide is too far past the focal point. So what's going to happen is our image that we're seeing in the microscope is going to look empty because there's going to be interference with other waves of light. Okay, so it's going to look like we deleted the microbe from the image, and this is what we see here. We have these empty spaces. Okay, now this all is assuming that our microbe is horizontal to the slide. Okay, so it's directly in the same plane. But what if our object is in the different direction and our focal point only hits a part of the object? What's going to happen is part of the object is going to be in focus and the other part's not. Now you can see this here with our image. So these 2 segments right here are in greater focus than these 2 segments. And that's because our specimen is going through in a different direction in the focal point. Okay, so one other thing we need to talk about is depth of field. So depth of field refers to how much of the specimen that you can see underneath the microscope at a time. Okay, so the more we magnify in, our depth of field is going to change. In fact, our depth of field is going to get smaller. So we're going to see less and less of the whole specimen and more of a specific part of the specimen. So our depth of field decreases as we increase magnification. Now, if you've ever used a scope on a rifle, this is what happens. You're going to find that deer in the trees, and you're going to zoom in, and you're going to start to see less and less of the surroundings around the deer, and maybe just the whole deer. And maybe if you're focusing in on around the heart or between the eyes, wherever you want to shoot the deer, you're going to focus in until you only see that specific spot where you want to shoot. Okay, that's an example of our depth of field decreasing. Which brings us to our last slide. The last slide's really cool. There's this group out of UC Berkeley who has designed a microscope stand that can be used with our cell phones so we can have a traveling microscope. Now, microscopes are used a lot in the medical field as a diagnostic tool. What are some of the benefits of a microscope being a diagnostic tool? I'll give you a second to think about it if you want to pause the video. Well, if we have a microscope on hand, we can see certain microbes, and being able to get a visual of those microbes will help us diagnose what the patient has more quickly. Now, microscopes aren't something that are available in every country in the world. In fact, a lot of third world countries will have them in the hospitals in big cities, but not in rural areas. So what comes in with this, what we call the cell scope, okay, that group in UC Berkeley 3D printed a microscope stand. Okay, so it has a place to hold your phone. It has a lens that you can, and a slide holder that you can put on top of the lens. And that lens can hook to your phone. And then it has a place for this pen light to give us that light source for our sample. So what happens is when we do this, this will change the photo app on your phone into a microscope. So we can look at different tissues and use our phone to diagnose certain diseases. Some of the limitations though are our phone only has so much resolution. We can only magnify so much. So we really can't see really small microbes like bacteria, but we can see bigger ones like the eukaryotic microbes like the microbe that causes malaria. We could use this for. Okay, but we can't see everything, but we can see enough to diagnose some of the more common third world diseases that we come across. So malaria and a couple others. Okay, if you want to learn more about CellScope and what they're doing with this project, there's a really cool one-page write-up about it in our textbook in 3.3. Okay, so that is section 3.3. We now know what a compound microscope is. We know how light shines through a sample in light microscopy. We've learned about focal points and how to focus it. We've learned about the different parts of the compound microscope. We have the objective and ocular lenses. We know how to calculate our total magnification. So you guys are really starting to have the basics that we're going to use in the lab. Now, 3.3 is going to go into 3.4, and 3.4 we're actually going to go into how we stain and prepare samples on the slides to view in lab. Okay, so we'll see you for section 3.4.